So next we're going to hear more about imprinting from Dr. Salter's fellow laureate, Professor Azim Surani. And Professor Surani is the Director of Germline and Epigenetics Research at the Wellcome Trust Cancer Research UK Gairdner Institute and Marshall Walton Professor at the University of Cambridge. Dr. Surani, as you heard, along with Davo, was really the first to identify that genomic imprinting exists, and he's certainly continued to work on genomic imprinting, but his more recent work has really moved on from that to focus on the genetic basis of germ cell specification in mice and humans, the origin of germ cells in the embryo, and their derivation from stem cells in vitro. So please join us to explore more about genomic imprinting, Professor Azim Surani.
Thank you very much. It's a great honor, and I want to congratulate Davo Salter, my co-recipient of this wonderful award, Gairdner Award. Davo actually explained to you really beautifully what genomic imprinting is. And when I was actually a PhD student, I was also interested in this question. And the question that I was interested in at the time is, are both parental genomes essential for mammalian development? And I was working with Matt Kaufman a lot on trying to understand how the parthenogenetic embryos develop or fail to develop. Now, I'm not going to talk about that, but I think what I just want to point out is some of the impact of the discovery of genomic imprinting that Davo Salter just described to you. So the question that we were asking, and the answer is that, in fact, in mammals, both parental genomes are essential for development because they inherit this epigenetic imprint marks from the germline, and these marks actually endure and into adulthood. So you retain these marks even now in all your cells if you look at them. And the key function, they form some kind of a network, and the key function they have is to regulate mammalian development and physiology. And we know that if imprinting goes wrong, you can get various human diseases. But I think that one of the key contributions from this phenomenon has been to the field of epigenetics, which is a very wide and rapidly growing field now, and very important for many diseases, including cancers and so on. So from this studies that we were doing, it was obvious that we really needed to look at the germline because a lot of the key aspects of the mechanisms were happening in the germline. So I want to take you through some of the work. And so we know that there is information coming from the germline in the form of imprinting, and that's the epigenetic information. There are other bits that are also transmitted, like mitochondria, from the female germline. Now, if you look at the development from the zygote through, the primordial germ cells themselves are formed very early in the pre-gastrulation embryo. And after they are specified, they start to migrate towards the developing gonads. And during this migration, they go through a very profound epigenetic resetting process, which partly involves erasure of imprints that exist before new imprints can go on. This is probably also the time when the epigenetic clock is reset. So this idea that I'll just put forward is that you have an epigenetic clock which measures your epigenetic time, and you can compare it with your chronological age. And if your epigenetic clock is going faster than your chronological age, it means that you're aging faster than you should be. So this part of the development of the germ cells is extremely important, and it's an area that many people have explored. And then there is also the transmission of a large amount of repetitive sequences that are transmitted. Maybe 70 to 80% of what is transmitted to subsequent generations is non-coding. It's really repetitive elements. And in humans, a lot of this is hominoid specific. So I want to describe now, tell you about the mechanism of how germ cell specification actually works. And I'm going to first tell you about the mouse work which was done. So before we started looking for the genetic basis for germ cell specification, there was work done by Lawson and also by Bridget Hogan looking at the signaling and the origin of primordial germ cells. So they come from this very proximal part of the post-implantation epiblast. So that was known, but what was not really known when we started this work in 2000 was what is the actual mechanism, what are the key genes that are involved in specifying germ cells. We know that at day 7.25, there are about 30 to 35 primordial germ cells already sitting there specified. So the experiment that we did together with Sheila Barton and Mitinori Sayutu was the following. So what we did was we took these 7.25 embryos and we dissected out this fragment consisting of about 300 cells together with the 30 primordial germ cells. And we ended up with 83 single-cell cDNAs. This was very early days of single-cell technology, so we were not at all sure. In fact, the reviewers, when we put in for a grant application, were not sure this was going to work. So we did this screen, and we ended up with 10 cells we thought were primordial germ cells, and then did a screen, and it turned out that the screen worked, and we started to pull out some genes. Stella was the first one, and then Fragilis, and subsequently BLIMP1. And I think this was really one of the key experiments which opened up the field of mammalian germ cell biology. And a lot of the work has been done by many people, our lab and other former colleagues and other labs. And to summarize what we know now is that you require 3 key genes for germ cell specification in mice. There's a PRDM1, or sometimes called BLIMP1, PRDM14, and AP2 gamma. So these are necessary and sufficient to start up the germ cell lineage. And these 3 factors form 3 key functions. The first thing they have to do is to suppress the somatic program. And there's been a lot of work being done to show how this works. They also upregulate the germ cell program. So the germ cell specification program starts while there is repression of the somatic program. And the third function, which is also key in this, is the initiation of the epigenetic program that I told you about. So this actually established the basis, the genetic basis for the specification of mouse germ cell lineage. And from this, as Davor was pointing out in his talk, since the discovery of these genes, we've now reached a point, quite rapidly, surprisingly, that we can now go from understanding the genetic basis of germ cell specification to making synthetic gametes from reprogrammed skin cells. So this is actually quite remarkable, actually. When you think about it, this is quite a profound change in the way we think about soma and germ cells. And that's because this actually breaks what's called Weissman's barrier. So Weissman was a real pioneer in the field of germ cells. And he basically, just to give a brief flavor of what he was saying, is that there are 2 types of cells. There are somatic cells which will perish with each generation, and germ cells that are immortal because they theoretically give rise to endless generations. And the epigenetic resetting that I was talking about, the resetting of the clock, is very important to ensure that the germ cell lineage remains so-called immortal, gives rise to generation after generation. So this epigenetic resetting is quite pivotal to this. And as Davor also pointed out, one of the outcomes from these studies and advances that have been made is that it's possible to convert somatic cells, skin cells, to viable gametes via induced pluripotent stem cells. So you can make this work done by Hayashi and Saito, and you can see that we can cross from somatic to germ cells, which is why we call it breaking Weismann's barrier. There was another unexpected outcome from these early studies that we're doing, and that was a technological advance, which was the single-cell RNA sequencing. So when we were doing this work, we were approached by Applied Biosystems, Kai-Kin Lau, and he thought that it should be possible to do to make single-cell transcriptome. And the very first paper was published in 2009 on trying to make this single-cell RNA-seq, and published a number of papers at that time. And since then, you can see the number of technological advances that have been made on single-cell analysis, the resolution of single cells. And in fact, there is now also a big project on human cell atlas, which is to try and get the transcriptome of every single cell in the human body. So returning to the germ cells specification, and what happens is that after they are specified at day 7.25, I told you that they start to migrate towards the developing gonads. And while they're migrating, they go through this epigenetic reprogramming. And this, I won't go into the details of it, but this does not happen by a single mechanism. There are parallel redundant mechanisms that are operating to ensure that the epigenome reaches the ground state, which is that the levels of DNA methylation go down to about 2.5% or less. So this reprogram is very important. It also ensures that if there are epi mutations, they will be erased. So this is like a computer disk. You clean it up before the genome is passed on to the next generation. And the mechanisms involved are multiple and parallel. It's not a single enzyme that does it. So that was the work that was going on for over 10 or 15 years, and now still continuing to try and understand precisely what happens. So I want to now move on to the work on the human germ cell lineage. We know quite a lot about the mouse development and mouse germ cell lineage, and so why would we want to look at human? And I think we have learned quite a bit about the basic principles of mammalian development from work on mice. But the reason why it is important to look at humans is because although the development of embryos looks very similar between mouse and human, there are molecular differences between how precisely these events take place, which lead to the formation of the establishment of the pluripotent stem cells, pluripotent cells in the embryo. But the other thing which is obvious is that you can see that mouse embryos develop as X cylinders. Human embryos develop as bilaminar discs. So the actual embryo doesn't look the same when you compare mouse and human. So now there are lots of technical and ethical challenges if you want to look at, for example, germ cell specification, which occurs we think on day 17. And so the regulations don't allow you to work beyond day 14 currently. So we have to find other ways of working on this. So one obvious way of approaching this is to try and use human embryonic stem cells using reporters like nanos3, which are very germ cell specific, and try and make germ cell-like cells from that. So that's what we tried to do. And just so that we have something to compare them with, we used germ cells from aborted fetuses, so we could compare these that we make in culture with the real thing. So we had a number of controls and so on. And this is what the actual experiment looks like. So you can take these epiblast cells, and you can use a variety of cytokines to drive them towards mesendoderm, towards the gastrulation end of development. And what we were doing was to take these cells at 6-hourly intervals and to see at what point they become competent for germ cell fate. And this we can test by exposing them to BMP, which is the signal for inducing germ cell fate. So this takes 24 hours, and midway along this journey of these cells, we saw that you can induce human germ cell-like cells because we had this reporter. But this occurs transiently. It's a transient state. The competent state is transient. They get there, and then the state disappears before they go on to the next phase. Now, we can study this in great detail because it's a very simple two-dimensional model, and it's very reproducible. We can use single-cell technology to understand how cells become competent and lose it. So we can do a number of studies. But the key thing that came out from this study when we started looking at these in detail is that very surprisingly, SOC17 turned out to be the key gene specifier of human primordial germ cells, which is not the case in mice. And this is surprising because SOC17 later on is a primary inducer of definitive endoderm. So it's the same transcription factor, does very different things in a very short space of time. So we can now explore this further. And what we can also show is that if you have just two factors and induce expression of these two factors in competent cells without any cytokines, you can induce very large numbers of primordial germ cells. The dosage is important. We don't understand exactly how, but the gene dosage is very important. So in order to understand a bit more about the mechanism, we can now ask what are the targets of these two factors, which seem to be very critical for germ cell fate. And these are just the chip-seq type experiments, and you can see that the binding of BLIMP1, which is one of the factors, compared to SOC17, there's very little overlap. So in other words, they're not co-binding to specific sites, they're binding to different places. So that is one of the outcomes from this analysis. The second thing that comes out of this is that we find that SOC17 is primarily involved in the upregulation of the germ cell program, while BLIMP1, although it also upregulates some of the genes, like VENTX, for example, its primary role is to repress the somatic program at this time when germ cell specification is occurring. So we're looking at many targets and how they are precisely regulated. I just want to tell you about one of them, and it turns out that BLIMP1 itself is a target of SOC17. And we can see binding of SOC17 to the promoter region of BLIMP1 and the upstream putative enhancer region. So we think that this SOC17 is binding to these two particular sites, and we wanted to test whether this has functional consequences or not. So this is the kind of experiment you can do. So these are the two regions where the SOC17 is binding, and there are 3 SOX motifs in the enhancer-like region and 2 in the promoter-like region. So what we can do is to clone these out, and we hook it up, make a reporter construct with luciferase, and we can test the activity then and see how these regulatory elements respond, and by looking at the induction of the luciferase. So if you have no SOC17, you get no signal. But if you express SOC17, you can see very high signal of luciferase, which means that these two sequences are perceiving SOC17 as the inducer of the activity. Now we have introduced mutations in the SOC17 motifs. So if you put a mutation in the enhancer-like region, you get some decrease in the luciferase activity, but you have a much stronger effect if you have a mutation in the promoter region. And if you combine the two together, you get a very strong repression. So all of this, this is telling us is that indeed SOC17 is binding to this particular target and all the other targets that we have, which drive the germ cell program. And we can try to understand the actual mechanism of how the induction of germ cell genes can work. So obviously the SOX family is very important. So we have SOX2, they belong to different subfamilies. So SOX2 is very important for pluripotency and for the formation of the neuroectoderm. And SOX17, which belongs to F subfamily, is very important for endoderm. And now we also know that it's important for specification of human primordial germ cells, but not mouse. We also know that for SOX family members, they require partners. They don't work alone. They have partners. So in the case of pluripotency, SOX2 combines with OCT4 to pluripotency genes, which maintains the pluripotent state, for example, in the pluripotent stem cells. And we think that what's happening is that SOX17 is, OCT4 is also important as a partner for SOX17 to drive the germ cell program. And what is interesting is that as soon as you start to induce germ cell fate, there's repression of SOX2, very rapid repression of SOX2 by two different mechanisms, which I won't go into. But what that does is it releases OCT4, which can now partner with SOX17 and go and drive the germ cell program. So here's one factor which is swapping. And we don't understand exactly all the mechanistic bits, but we're trying to get at that now. So SOX2 OCT4 are critical for regulating pluripotency, and OCT4 switches partners from SOX2 to SOX17 doing germ cell specification. And we are wondering whether SOX17 acts as a pioneer factor to initiate enantiosomes for germ cell program. So this is just a model at the present time. We are working through this to see to what extent this model is correct. We have also done motif analysis, so we can see here that we can look at the motif for where the SOX17 peaks are and look for cofactors. So the cofactors for the germ cell genes are OCT4 and this other factor, TFAP2C. These are two very important for driving germ cell fate. And the other interesting point about this is that I told you that SOX17 little later on will induce definitive endoderm. And if you look at the partners of SOX17, they're completely different. So it doesn't partner with OCT4 for the specification of definitive endoderm. There are other genes like FOXA1 and so on. So we can see the logic, the molecular logic of how a transcription factor, one single transcription factor can induce two very different cell fates. And so one of the hopes is that this very simple model, two-dimensional model we have, may allow us to get very deep into the molecular mechanism. So we are doing other kinds of experiments where we can try and ask about more developmental questions, about the origin of the germ cells and so on. But for the molecular analysis, we can try and look at this in this way. So of course there is also epigenetic resetting to the ground state for human, because this is much more difficult to study than in mice. We can get material from aborted fetuses, so we've been collecting material. We've collected all the material over the last year, which are already and being sequenced now. So what we want to do is to try and look at all the histone modifications, the DNA demethylation events that are going on, non-coding RNAs. We're also looking for to get information about how this program is working in vivo. And we are hoping that we'll start to get the first glimpse of it towards the end of this year. The second question, which is of interest to a lot of people, including our group, is that this primordial germ cell that we make in culture from stem cells are equivalent to about 2.5 to 3 weeks. And we'd like to see if we can take them further. We have succeeded a little bit, but we're still far away from our ideal. So our first objective is to get the equivalent around 7th week, because this is the period of which this epigenetic resetting is occurring, and we cannot study this without a really good in vitro model. And so that's what we're trying to do, is to try and get an in vitro model for this human germ cell development beyond the specification stage. While this, we know that the epigenome here, or the methylome, gets demethylated just in the mouse. So if you look at germ cells at this point, they're acutely hypermethylated. The levels of methylation goes down to about 2 to 5%. So they go through a very similar thing. But we think that the mechanism that initiates this program is different from the mouse, because we now have a SOC17 there, which would be involved in the initiation of this epigenetic program. So that's something that we're trying to look at while we're looking for targets of the SOC17 and BLIP1. Some loci are resistant to epigenetic reprogramming, so we are particularly interested in the transposable elements which have come in later in the primate lineage, in great apes from the development of evolution of gibbons. So that's about 30 million years ago. So this is a huge amount of non-coding DNA that is being transmitted transgenerationally over millions of years. And when we did this analysis, we could see that the resistant loci that we find that are not reprogrammed are primarily, majority of them are very early transposable elements. And one of the tantalizing things about this information that we have is when we do bioinformatics analysis on this, it suggests that they might have some role in neuronal development and metabolism. So that's the bioinformatics. And that if you do genome-wide association studies, there's also a suggestion that they might have some implications for neuronal disorders. So this is bioinformatics. Unfortunately, I don't have time to tell you, but I can say that we have been thinking about this over the past 2 years, and we're starting to do some work on it now. And we are looking at some of these transposable elements. In particular, one particular one is the LTR12C, which when we look at human fetal brain at 12 weeks, we see a change in the epigenetic signature from methylation to histone acetylation mark, which is a mark, which is one of the marks associated with enhancer elements. So we are wondering whether some of these repetitive sequences have undergone mutations, have acquired transcription factor binding sites, and may have been co-opted as regulatory elements. So that's one of the ongoing, quite difficult projects we are trying to undertake, to see if some of these repetitive sequences, which are transmitted over 20 or 30 million years, have been used. I think this would be quite interesting, and perhaps, speculatively, you might think that one of the big differences between chimps and humans is the brain, the size of the brain, and whether some of these coding DNA, which is being transmitted, has somehow been co-opted and has been important for the evolution of some of the human-specific features, including the human face, which Jenna Weishocker is working on the neural crest cells. So those are some of the things that we are working on. So this is the kind of the summary of what I've told you. So we start with sperm and eggs. They come in with imprints. There is some demethylation earlier on, but as the embryo develops and forms epiblast, it acquires more DNA methylation. The somatic cells retain most of this DNA methylation as they differentiate. Those that opt to go for germ cell fate, they start to erase DNA methylation. I told you about getting down to the ground state. But there are some single copy and repeat sequences which are resistant to reprogramming. And we are wondering whether they might have some role. And then, of course, later on, as germ cells develop, they acquire new imprints, the initiation of imprints. So you see the whole cycle is completed. So I think this is potentially a very exciting time to get into human development, which we actually know relatively little about. And so now I think there are opportunities to think about looking at how the early human embryo develops. There are ethical challenges and technical challenges. You need to look at the origin of primordial germ cells in human embryos in some way, and to understand the epigenetic resetting and inheritance. We are very interested in this transmission of these resistant loci and whether they might have some function. And we have a collaborative project with Patrick Shinnery to look at mitochondria, because there's a mitochondrial bottleneck in very early germ cells when they go through purification event, when some of the defective mitochondria eliminated or the cells with defective mitochondria eliminated. And this is hugely important because of its implications for human diseases. And so the model that we are developing will provide us with opportunities to look at some aspects of mitochondrial biology. Now, way into the future are these questions about synthetic gametes. We know, I told you can make them with mice. They're all theoretical. At the moment, there's also the question of germline gene editing, which is also an important area. And of course, we're not anywhere near there, but I think this is the right time to start to debate these issues because we don't know how rapidly these advances will occur, and we want to be prepared for it. And now Janet is actually contributing a lot to the debate on gene editing, and I think it's important to inform the general public about some of these advances that have potentially quite huge implications for the future. So this is the final slide. I can't, unfortunately, put all the names of people who have worked in my lab. There have been huge numbers, and I think they know who they are. I mentioned some of them. But the contributions in my lab and subsequently after that, has been enormous. This is my current group. I'd like to thank Wellcome Trust, who have funded me generously over many, many years, and others, MRC, BBSRC. And I have currently some really wonderful collaborators and going through to the next phase of an exciting journey, I hope. Thank you.